Awesome. Okay, so welcome everyone. If you're here for the Science by the Sea seminar, you are in the right place. We have a set of 3 really great talks ready for you today. My name is Maya Waltham. I work at Hawaii Sea Grant as the Assistant Director for Research and Fellowships. And again, we're really excited to have you here for the Science by the Sea seminar series. In the months of November and December, we're featuring our Sea Grant graduate fellows and our PICAST graduate scholars. PICAST stands for Pacific Islands Climate Adaptation Science Center. So these graduate students are supported by Sea Grant and PICAST to conduct research on oceans and coasts. They're going to share some of their research results, some of their progress today. And in addition to pursuing their graduate degrees, doing this cutting edge research, they receive training from Sea Grant and PICAST in science communication and outreach. And we ask that each of our graduate students completes 50 hours of outreach a year to the broader general public. So today we're going to hear 3 different talks. First, we'll hear from PICAST graduate scholar Becca Lensing. Her talk is titled, "There Was an Island That Swallowed a Rat." Then we're gonna move to our second talk. It's from 2 Sea Grant graduate fellows. Their names are Matt Duff and John Cameron. Their talk is titled, "A Cesspool Story, How OSDS Has Influenced the EVA Plain." And we're gonna round out the talks today. The third talk will be from Sea Grant graduate fellow Zack Rago. His talk is titled, "A Cage Match for Seascape Ecology." So if you've joined us before, you know that we are asking for questions in the chat as we go along. We're going to take a question in between each of the speakers, and we have time saved at the end for a longer Q&A. So please send us your questions as we go along. And some other housekeeping logistics kinds of things is we are recording the seminar today. There are some folks that weren't able to participate in real time, so we're offering for people to visit the recording on their own through the Hawaii Sea Grant YouTube channel. Okay, so I'm going to stop sharing my screen and we'll call Becca to the Zoom stage. Becca, whenever you're ready, the floor is yours.
All right. Maya, thumbs up. You can see my screen.
Yes, we can see your screen. It looks great.
Great, thank you. So yeah, my talk today is about an island that swallowed a rat. Maya told us that she wanted us to tell a story, and I really ran with it. And in lieu of some results, we're still waiting on some sequencing results as due to postponed due to COVID. I have actually decided to tell you 2 different stories. In my work, I'm always thinking about invasive species. My particular project focuses on one of these invasive species I'm going to tell you about today, but we also spent a great deal of time with the other invasive species while in the mangrove forest doing some of our field work. And so every time I think about invasive species in Hawaii, I am always reminded of an old nursery rhyme. You might not have heard of it before. I find a lot of people haven't heard of it, but the nursery rhyme is called, "There Was an Old Lady Who Swallowed a Fly." And it goes something like this. There was an old lady who swallowed a fly. I don't know why she swallowed that fly. Perhaps she'll die. There was an old lady that swallowed a spider that wiggled and tickled inside her. She swallowed the spider to catch the fly. I don't know why she swallowed the fly. Perhaps she'll die. There was an old lady who swallowed a bird. How absurd to swallow a bird. She swallowed the bird to catch the spider that wriggled and tickled inside her. She swallowed the spider to catch the fly. I don't know why she swallowed the fly. Perhaps she'll die. There was an old lady who swallowed a cat. Imagine that. She swallowed a cat. She swallowed a cat to catch the bird. She swallowed the bird to catch the spider that wriggled and tickled inside her. She swallowed the spider to catch the fly. I don't know why she swallowed the fly. Perhaps she'll die. There was an old lady who swallowed a dog. What a hog to swallow a dog. She swallowed the dog to catch the cat. She swallowed the cat to catch the bird. She swallowed the bird to catch the spider that wriggled and tickled inside her. She swallowed the spider to catch the fly. I don't know why she swallowed the fly. Perhaps she'll die. There was an old lady who swallowed a goat. She swallowed imagine that. She opened her throat and swallowed a goat. She swallowed the goat to catch the dog. She swallowed the dog to catch the cat. She swallowed the cat to catch the bird. She swallowed the bird to catch the spider that wriggled and tickled inside her. She swallowed the bird. She swallowed the spider to catch the fly. I don't know why she swallowed the fly. Perhaps she'll die. There was an old lady who swallowed a cow. I don't know how she swallowed a cow. She swallowed the cow to catch the goat. She swallowed the goat to catch the dog. She swallowed the dog to catch the cat. She swallowed the cat to catch the bird. She swallowed the bird to catch the spider that wriggled and tickled inside her. She swallowed the spider to catch the fly. I don't know why she swallowed the fly. Perhaps she'll die. There was an old lady who swallowed a horse. She's dead, of course. So in that particular fashion, in sort of this unusual storytelling, I'm going to be telling you about, two separate rhymes of different cadences about two invasive species and their story here in the Hawaiian Islands. The first is about rats and the second is about mangrove. So without further ado, there was an island that swallowed a rat. There was a ship that swallowed a rat. It crawled aboard in England, thought not but a sprat. The ship set sail to circumvent the world, but oh, from its travel, what tragedy unfurled upon the people, culture, Lo'i and sand flat, for the island that would swallow that rat. There was an island that swallowed a rat. It ate the island sugarcane, it got so fat. It multiplied and spread from the ship's manifesto, spreading rat, lungworm, toxo, typhus, and lepto, and a bubonic plague of death and black brought to shore on the rat's flea-covered back. There was an island that swallowed a flame that raged out of control. What a shame. The island swallowed the flame to catch the plague. With unknown dead, the numbers vague. The flame leapt across the streets of Western downtown and in 17 days raised 38 acres of Chinatown. 4,000 homes, so much displacement begat, all because of that one flea-covered rat. There was an island that swallowed a mongoose. Imagine that, intentionally put there and let loose. The island swallowed the mongoose to catch the rat. It ate the island sugarcane, it got so fat. Introduced to keep the rat population kept low, they also spread rat, lungworm, toxo, typhus, and lepto. Added to the problem these mongoose did, so of their captive breeding the state forbid. But there was another problem with mongoose, you see. Rats are nocturnal, their sleep patterns disagree. Not so much were eaten, as that was not their way, rather the eggs of turtles and birds, the precious nene. Greatly endangered these animals became, with the mongoose more than partly to blame. Hindsight is always 20/20. History will always have mistakes aplenty, but sometimes the solution is no better than the predicament. We can turn a blind eye to history to our detriment, or we can build back better and see a future in that to help save the island that swallowed a rat. That is the conclusion of the rhyme. But there are some efforts that you personally can take to reduce some of the negative effects of rats in your area. That would be rodent proofing your home, reducing all the areas that they can come through, and regular sanitization so that they don't have excess food to eat, as well as trapping. It's always best to put the traps where the rodents are and bait it with various foods or even bed stuffings that the rats might use to create their nests. And of course, if you need any further information, there are all these handy resources from the Hawaii Department of Health over on the left. And with that, it brings me to our second story today. This is one that I am much more closely and personally related to because this is what my research focuses on. We do studies on the south shore of Palau, on Moloka'i's south shore, that is, as well as in Kaneohe Bay in He'eia Fish Pond, where actually this photo was taken. All right, so the story of mangroves in Hawaii goes something like this, and I set it to the cadence of 'Twas the Night Before Christmas, sort of being in the holiday mood. 'Twere islands in the sea, specks in the Pacific. Agriculture was changing, substantial and prolific. Sugarcane replaced lo'i ponds and kalo. Sediment ran off streams, straight and narrow. Pineapple farming grew with great fervor, and runoff worsened to the keen observer. Precious topsoil was lost from the land, but worse was the reef, smothered out of hand. To save the reefs, the people looked to one little tree, but introductions failed from out of the Indian Sea. When out from Molokai, there arose a success, a species from Florida that would grow nonetheless. It sprang forth with a vengeance, this red mangrove did. Through streams, ponds, and flats, it rose up unbid. Trapping sediment, as was its goal, the lack of natural predators soon took its toll. Fish were blocked on their routes toward fresher water. Pathogens, trash, toxins accumulated from runoff wastewater. Burrowing animals were choked of oxygen and fled, and invasive species moved inside and made their bed. Mangroves were introduced to protect the reef, but what would protect the coast from the toxic leaf? The trapped sediment built up to swallow local ia, so much history buried under this previously great idea. Communities sprang up to say, ku ho kua pa, gather the pohaku, the koa, grab your saw, for we can build a better future of sustainability for ourselves, our children, and the land's fertility. And away they charged with everyone who heard, citizens, scientists, and most importantly, the stewards. The fight is not over, not even close, but from these efforts of movement arose. Malama ka'aina, care for the land that feeds, because to restore the land is to restore our needs. And don't forget how mangroves became a blight. Happy holidays to all, and to all a good night. And with that, I would like to encourage everybody, if they are able, to do something personally that you can personally do to help these particular environments that are so suffering due to the spread of mangrove. You can volunteer and actually help to remove mangrove from these areas. Of course, I will always advertise my personal favorite, Pai Oheia. Also, a few more land-based, Kakao Iwi always accepts volunteers. They have started back their community work days on a socially distanced, masked, and reservation basis. So always feel free to reach out to them and see how you can help. Otherwise, you can go and find your local fish ponds and ask how you can help and volunteer. Yeah, otherwise you can donate if you're not personally able to. All of these pictures, by the way, I would like to note are absolutely pre-COVID. All right, and with that, I would like to thank the partners that have been instrumental in our research project thus far. Members of Alagata Lab, particularly shout out to Rosie, Nalani, and Kui. We made a fantastic field team, all of us girls, out on Molokai. We also like to thank our local Molokai support, Clay Ching and his son Josiah, which helped us with transportation and cutting of a lot of mangrove to get to our sampling sites. Jason for his input and comments on how to best go about it, as well as to Daviana and Uncle Noah for their support and on-island logistical support. All right, Maya, I know I finished up a little bit ahead of time, but at this point I will take questions.
Thank you, Becca. That was excellent. A round of applause for you. If the audience has questions for Becca, please send them to us via the chat. I really love our graduate students are so creative and amazing. Becca, that was really cool to see you tell this story through that format. Awesome. Okay, I do see questions coming in through the chat. Rachel Lentz, would you like to ask your question out loud?
Sure. Yeah. Just this morning, I actually saw, can you hear me? My computer's closed.
Yes.
I just saw a really interesting story about how in the Caribbean, there are corals that are actually growing on mangrove. root systems, and that they have found that they're actually more resilient when bleaching events are killing off corals on the reefs, that these ones on the mangroves are actually surviving and not bleaching. Have you ever actually seen corals here growing on mangroves? I'm just curious.
Oh no. The invasive mangrove forest is a very different habitat than the mangrove forest in their natural ecosystems. So I always like to equate the Hawaiian mangrove forest to like Dagobah Swamp from Star Wars. The sediment is incredibly built up. It's very dark because there's a lot of shade, and the water is super turbid. It's like chocolate milk almost. So we've never seen any corals grow. I personally haven't seen sponges grow. I do know that there are species of invasive algae. I want to say it's Grilla oga that grows very prolifically around the root system. So no, we have not seen that in Hawaii, although, yeah, that was a great question. Thank you.
Thanks, Becca, again for your talk. There are some other questions for you in the chat, which you're more than welcome to continue the conversation via the chat box, or we'll have time at the end for a longer Q&A. What I'd like to do now is I'm going to bring Matt and John up to the Zoom stage and ask them to start their talk.
We should be good, I think.
We can see, it's not quite in presentation view yet. That looks great.
Cool. So thank you, Maya, and thank you guys all for joining us this afternoon to hear a little bit about our research. John and I are working on a project looking at groundwater pollution in the Ewa Plains region of Oahu, and particularly how submarine groundwater discharge, or SGD, can potentially be carrying nutrients and pollution and contaminants into the coastal regions, impacting coastal and marine environments. Sorry, John. We also just want to thank the Hawaii Department of Health, Hawaii Sea Grant, and the Department of Earth Sciences for helping us with all our work. So the primary issue that we're looking at with OSDS, which is on-site disposal systems, which is essentially like septic tanks and cesspools and things like that. And we're looking at how cesspools could potentially be allowing nutrients and contaminations to enter the groundwater and then make their way into the coastal regions via SGD. And so Ewa cesspools, they're a major concern because of that profound risk that they pose to nearshore marine waters. This map up on the top right, it's showing basically just an OSDS density on the entire island of Oahu. And our study region is within that red circle. And you can see that how red that area is. It's just as, if not more dense than any other area on Oahu. And so understanding how and where these nutrients are coming from is essential to protecting our coastal ecosystems. And so cesspools in Hawaii, they can release up to 53 million gallons of untreated wastewater a day. And for those of you who don't know much about cesspools, they're essentially shallow underground systems that dispose of sanitary waste. And although the structures can vary, they're mostly consisting of concrete cylinders that have an open bottom. And so the waste from things like washing machines, toilets, and sinks, they flow in through a wastewater pipe, and the solids will sink to the bottom. And then nutrient pollution from the sides that are percolated will flow out and can then enter the underground system and potentially get into the groundwater. And so also with the fact that Ewa's population is growing very fast and it's expected to increase by 71% by 2035, that's going to continue to increase stress on the current infrastructure and continue to exacerbate the problem. And so a little bit more background about the Ewa Plains. It was primarily used as sugarcane and agricultural land for decades. And then in the 1980s is when we really started to see a population rise and housing projects started to take shape. And so EVA is now the fastest growing region on Oahu. And this map on the far right, there's a lot going on in it, but really the most important part is within the purple border you see there, that is our main study area. And so all of the individual purple dots are OSDS systems, and where you can see the black, that is areas of OSDS that are so dense that they just can't even be distinguished on a map. And so it's really showing how many OSDS systems are within this very small area. And so we're really interested in seeing how having that many OSDS sites like cesspools could potentially be impacting the coastal reefs and the coastal waters. And a little bit more specifically about the numbers of OSDS that are in that area, there's 1,373 OSDS in EVA, and 1,246 of those are cesspools. So the vast majority of everything you see there is cesspools. And the EVA plain in general, it's a very large and dry plain, very little topography. There's no perennial freshwater streams, and it gets very little rain. And the geology is unique in that it's dominated by limestone deposits and karst topography. And so the picture down in the bottom there, that's a karstic cave. And karst is really, it's a type of underground topography that's formed by the dissolution of soluble rocks, particularly limestone. And this can make landscapes like this particularly more susceptible to contaminants because sinking streams, as you can see in this diagram here, there's that stream is sinking down into the sinkholes, and it's providing a rapid route for unfiltered contaminants to then make its way into those kind of nooks and crannies and then escape from the land into the ocean waters as contaminated SGD. And so this figure right here, this is just sort of a simpler version of the figure we just showed, where it's just showing the OSDS. And so you can see here, even in this zoomed in shot, there's still that black smudge there where that's just so many cesspools in OSDS that you can't even distinguish the individual ones. And so there's just a very large amount in a very, very small area of cesspools. And so in terms of the specific work that we're doing to try to determine what types of contaminants are entering the system and what risks they could pose, we're looking at taking water samples in both the wet season and dry season to determine if there's any seasonal variability. We're doing pore water samples from beaches. We're focusing on 4 main beaches, 2 of the beaches, Puuloa Beach Park and Oneula Beach Park, we're using as our sort of priority sites because those are closest in proximity to all the cesspools. And then Kalealoa Beach Park and White Plains are sort of our pristine sites, which are located outside of the priority zone. We'll also be taking samples from wells that are located throughout the EVA plains and also taking algae samples. And so we're specifically looking from those samples, we're going to be looking at the salinity, the nitrate and nitrite. We're going to be looking at some certain isotopes like nitrogen 15 and oxygen 18. We're looking at phosphorus, inorganic and organic carbon. And the reason we're looking at all that is because we want to try to use all of it to get a clearer picture of where the contaminants are coming from, because sources like agriculture and wastewater from cesspools, they'll all have very distinct isotope numbers and nutrient numbers. And so by understanding what those numbers are in the water that we're collecting in the EVA plains, we can better be able to say, well, it looks like it's coming mostly from cesspools, or it's coming from agriculture, or it's coming from natural sources. And so algae is something that we're looking at in particular because it acts as a very good tape recorder for pollution. And that's because it assimilates nutrients from the water column and accumulates them in their tissues. And so the nitrogen values that we find in the algae can correlated with the isotopes and nutrients of water can really help us pinpoint where we think the sources of the contaminants are coming from. And so the 4 algae species that we're focusing on are Laurencia, Cladophora, Ulva, and Acanthophora. And the images on the left here, it's showing submarine groundwater discharge springs. And so the top picture is a freshwater spring that we found, and that was when it was completely covered in sand. And below that picture, that is that exact same spot, but that's after all the sand's been carved out. And so you can see how there's a crack that runs along that rock. And that's where we have fresh water that's coming up. And that's where we can get the freshest samples, which will be the best indication of where the contaminants are coming from.
All right, so this isn't the most visually beautiful map. However, it shows how many samples we've taken and where we've taken them from. So you have the red outline, which is that priority zone that we're focusing on, and then a couple different clouds. This cloud is all of our groundwater wells, including housing developments like Matt said, and then golf courses. Along the beach, you have a blue cloud, which is all of our pore water samples. There's about 22 along the coast of Eva right here. We've sampled some of them 4 times, 3 times, 2 times over the course of the last year. The stormwater canal is a drainage canal that's right along Fort Weaver Road in Eva Beach. And we sampled it because there is an output for it right at the shoreline right there. Eva doesn't get too much rain, so it's not as useful, but there's still freshwater input from the city into the water there. This is just another image of all those OSDS without that removed, so you can see how kind of the range of sample locations we have currently. So here are pictures of us collecting these pore water samples. On the left-hand side is our YSI, which we use for all water quality parameters such as salinity, pH, dissolved oxygen, and temperature. This helps us identify if it's groundwater or if it's just seawater. Seawater is 35 salinity, so anything under that number is what we shoot for because there is fresh water in it, and that's what we're looking for. The middle picture is me filling up a sample bottle. In this bag, it is not pictured, but there's a pump with a filter and a tube that leads to the right picture, which is our piezometer. And a piezometer is just a tube you put in the ground to suck up the groundwater with holes drilled in it with a screen, so it doesn't pull up sand, and you just get groundwater. Here are the other types of samples we got, the well samples. On the left-hand side, you see an observation well. The observation well is located at a golf course, and it's nothing more than just a hole that was drilled in the ground to test the groundwater and monitor for mainly salinity. Since it doesn't have a pump built in, we had to use this clear tube here, which is a groundwater bailer. We had to lower it down. It'll fill with water all the way up, and you bring it up, and you do that a couple times to cycle as if you were turning the pump on. And we collected several samples that way. On the right-hand side is an irrigation well, and these ones are much easier because they have a pump built in. So it's as simple as turning on a faucet, filling up our sample bottles, and then throwing them in the cooler, which is nice. Continuing with more photos, we're doing testing for fecal indicator bacteria, which mainly looking at E. coli, and E. coli is in the news all the time with produce and stuff, but it does appear in groundwater, and it's not an end-all be-all for septic tank and cesspool pollution. However, it's a very good marker that you can look for. So these are just pictures of us preparing the trays. The left-hand side is a prepared tray that's ready to be put in an oven for 18 hours and let the E. coli kind of mature and grow. The right-hand side is Matt preparing the water samples. There's a little powder that you dump in the water and shake it up, and it activates all the microbes in there that you're looking for. And when you take it out of the oven, it's probably the most fun part. It will look like this. The left-hand side, it'll turn yellow if there's any coliforms in there, and on the right-hand side, it will glow under UV light if there's any E. coli. One last set of photos, I think. This is how we see under the ground. On the left-hand side this is all geophysical resistivity. If you shoot a current of electricity into the ground, the rocks and water will give you different numbers. So the left-hand side will shoot a current through this cable on the right-hand side, through these spikes into the ground. And when you do all that, the outcome looks like this. And you can see the different layers of rock and water as you go down about 20 or 30 meters, which is very useful in finding springs and under other karst conduits. This is another map. These are actual results. These are algal tissue results and the isotopes that we're looking for, nitrogen. So you can see how these warmer colors, the reds and the oranges, indicate that there's more nitrogen in the algae that we collected from here, which is closer to the cesspools, than at here, which is our pristine beaches. And that could be a direct result of OSDS pollution. We don't have to spend too much time on this graph. It's just a graph, but it shows the same thing. The blue is closer to the cesspools. The red is the more pristine area. So there is some input from OSDS, we believe.
And so again, this is another graph that we don't have to spend too much time on, but it's a good indication of kind of what our results are telling us so far. And so this is comparing those two specific isotopes that we're looking at in water, delta 15 nitrogen and delta 18 oxygen. And so it's circled up here at the top. That is the two samples we have from freshwater submarine groundwater discharge springs. And so when you look at the oxygen and nitrogen data from those samples and compare it with this kind of handy chart that we have down in the right, where that chart is specifically showing you where if you compare the nitrogen and the oxygen, it tells you where you might expect the source of that nitrogen to be coming from. And so the SGD spring that we found, the numbers are almost perfectly in correlation with sewage and septic waste that you would find. And so, and those came from beaches that were in our close proximity to the cesspool sites. And so basically that is kind of leading us to think that our results are showing that cesspools and other human sources are indeed having some sort of impact on the groundwater quality and potentially the health of the Eva plains in general. And so thank you guys for listening. If there's any questions.
We also have our emails if anybody wants to email us questions that you think of later.
Thank you, Matt and John. A round of applause for your talks. Thank you so much. If there's questions from the audience, please put them into the chat box. And while we wait for those to come in, I'll ask a question. Are there any options for connecting to a sewer line in these areas of Eva with really dense cesspools?
Yes, all of Eva is on, if you're talking about like municipal sewer systems, all of Eva is on it. A lot of that big cluster that you saw is all the old part of Eva, and cesspools got outlawed by the state of Hawaii, but I don't think that will be changed until like 2040. So it's pretty expensive and not many people are doing it currently, but there's sewage for everyone who lives in Eva, but that was a recent addition.
Okay, any other questions from the audience? Thank you for your answer, John. Let's keep the questions coming through the chat box. We will have time for an extended Q&A at the end after we hear from all three speakers. So what I'd like to do now, I do see that Robertsley now has his hand raised. Would you like to ask a question?
There we go. Yes, really, really important work that you guys are doing. Those red dots around Oahu on your map. So there's been a lot of more of that kind of work ought to happen. And I have a particular interest in the Marine Life Conservation District in Pupukea, Waimea. I mean, thousands of people go there, and it would be nice if the government would monitor that so we can track trends, whether it's getting worse or better. But that kind of work isn't being done. Is there any chance you folks could expand the scope of your experiments and start building a data base in some of these other hotspots?
That's a good question. Our advisor, Craig Glenn, has had a couple of grad students who have done similar projects, North Shore, Eva. So our division is working on it, but it's kind of, they'll pick different areas. Eva has unique geology, which is why we're out there currently, but we want to expand even future grad students to get all the islands kind of classified and understood.
Thank you.
Thanks for that question. Okay, let's bring Zack Rago to the Zoom stage. And Zack, when you're ready, you can begin your talk.
Thank you, Maya. Okay, can we all see that?
Yes, we see your opening slide. Looks good.
Cool. Thanks everybody for coming. I'm Zack Rago. I work in Dr. Elizabeth Maiden's lab out here at the Hawaii Institute of Marine Biology. And my talk's titled "The Cage Match for Seascape Ecology." So I'm going to start by just jumping into who the participants are in the cage match. Why isn't that going? So the fighter in my scenario, fighter one, corner one, is our Manini, also known as Acanthurus triostegus. This is a very common reef fish throughout the Hawaiian Islands and throughout the Indo-Pacific. Very important ecological species because they are really important herbivores for our ecosystem. They spend most of their day just cruising around and chowing down on the algae that competes for space with our coral reefs. And we want more coral and less algae, so these guys play a really important ecological role. In the other corner, we have our challenger, locally known as the Roy or Cephalopholis argus. This is an invasive species. It's a species of grouper, was introduced here in the Hawaiian Islands and has become a problem because it gets blamed for a lot of the decreases in fish like Manini around the Hawaiian Islands. Basically, it goes around and it will eat anything that fits inside of its mouth, which can be quite large. And so this is our challenger in our cage fight today. And the prize is some good old Limu. In particular, I use Acanthophora spicifera. Again, this is also an introduced species, invasive, but was selected because the Manini actually will eat this, and that's kind of the goal of our cage match. So hopefully by now you're just asking, why a cage match? What are we talking here? Well, I'm really interested in predator-prey behavior. So by pitting these two up against each other in a cage match, we can actually learn a lot of really cool things about the broader picture that I study, and that is coral reef halos. So briefly, I just want to talk about what these are, what we kind of know about them, and what some of the hypotheses are as to why they exist. In all of these pictures, I hope you can see my mouse. You can kind of see we have patch reefs that represented by these darker areas. And essentially what we think is going on here with these really barren, bright rings around them is these are empty, barren areas of sand that are in between algae or seagrass in many circumstances and a coral reef. And the concept here, kind of denoted by this little figure at the bottom, is that the reef structure in the center actually provides protection for some of these small fish like our Manini or like our fighter. And they're only willing to travel so far away from that protection to go eat and feed on the algae before the threat of a roy or a shark or any other predator tends to outweigh what they're gaining and eating. So I'm really interested in finding out what some of the underlying mechanisms are that cause this pattern. And because we think herbivory and this landscape of fear is causing this pattern, or at least playing a significant role in this pattern, I've designed essentially a cage match to test out how fear impacts the grazing behavior of these herbivores. And the reason why I use a cage is because it's also called a mesocosm. And that's a great way to go about kind of looking at these large scale features like coral halos, because it brings a ton of balance to what we can do. On one hand, we can just do all field work, but that has limitations in the questions that we can ask, the control that we have in when we're studying something, versus the laboratory setting where we have immense control, but sometimes it can lack connectivity to the real world. So I use something called mesocosms, which are kind of this great balancing act between the two of those. I can create an enclosure that exists in the real world, but allows me enough control to be able to tinker with things and actually make little manipulations to the system to try and better understand what's going to happen in a predator-prey interaction, or what's going to happen in the behavior associated with that interaction. So this is what my setup looks like currently here at Coconut Island. And the way that I ultimately set up this system that I'm gonna share some very preliminary results with you is, I basically went out and caught Manini. It's very fun to catch Manini because we actually do it by going out in the middle of the night, flashing really bright flashlights into their eyes, and they kind of get stunned and you just scoop them up with nets. But this is how I set it up. On the left-hand side here was gonna be my control. And then I have 2 Maninis and 3 Maninis, and we're gonna basically throw in treatments and see how things react. Unfortunately for me, my third situation over there had a bit of an accident, and it seems that these guys escaped on me during my first round of experiments. So we're gonna focus on the 2 on the left-hand side. 1 control, and then 1 situation where I actually put in fake roids. In this situation, we're using laminated roy, which you can kind of see here. And this is the only photo that I included about, or actually within my experiment, because as you can see, it's not necessarily the clearest. And my actual study location has become colloquially known as Zack's mud pit. But you can see my fake laminated roy that's supposed to be scaring my, my Manini. And you can also see these little clips, which we're going to talk about in a moment. So rather than showing you really muddy pictures of what I spend my day swimming in, I thought that it would be best to actually show you a nice clear image. Basically, the setup here is I have this central structure that represents the coral reef. It's only accessible to my Manini. And then basically I have algae at 6 different distances away from that central protective structure. And then I've got 2 laminated roy. In my control system, there's no roy, so there's less fear involved with that situation to find out how there will be differences. And the idea is that in the control, we'll probably have a relatively large halo get created. They're going to be willing to go out farther. They have no fear. And in the situation that we have here with the actual treatment, it's going to shrink a little bit because these fish will actually have this fear influence upon them. So now let's actually check out what we are seeing in this situation. Oops, why is this not moving forward? Okay, apologies. So as we set this up, my first thing was introducing the Manini. They had about 2 days inside of these mesocosms or cage matches on their own, so I could observe them. They can get used to the system. And what we see them doing behaviorally before the experiment even starts is they tend to kind of go out and venture the exterior. They're trying to maybe find a way out. Whatever it is, they're very exploratory. They're not necessarily always sticking close to that central area. However, when we actually include the algae and introduce these fake groupers to kind of scare them and see how they react, their behavior changes a lot. They tended to hang out a lot at the central point, venture off a little bit and come right back and kind of do that throughout the day. So I was really excited. This is a really good start to what my expectations were for our experiment. And hopefully our results are going to show us the same thing. So now we focus just on the algae. Basically, I'm just measuring how tall is this little piece of Acanthophora every day for the past 6 days. In fact, this just ended yesterday. And I got some really interesting data. So they all started at the same height. This is essentially your day 0. And at the end of it, the average height looks like this. On the top, we have our control. We can see that they were eating a little bit very close to that central point, and they really didn't eat a whole lot as you moved farther out. However, in our treatment, we saw a lot more activity. They basically ate all of the algae at the first 2 distances, and they actually ate quite a bit of the algae at all 4 other distances. However, this doesn't really make sense. This goes against exactly what I thought would be happening. Our control should have this free-for-all of herbivory, and our treatment should actually be seeing a lot less because of the fear factor. So I was very confused by this, and I was really bummed out all week because I was like, this is just not going well. It's not doing at all what we expected. However, when I went back and really thought about how I had set this experiment up, I realized something that was happening, and it actually taught me really valuable information for when I move forward and actually take this experiment to another level. When we were catching our Manini, we attempted to catch the same sized fish across the board, but we actually weren't able to accomplish that. We only caught so many fish, and so we kept everything that we caught. And when I set this experiment up, I randomly put fish into each of these different systems. And in my treatment, I had one fish that was much larger than the others that I had caught. I would call it a subadult rather than a juvenile that the rest of the fish were. And so when I actually went back and looked at which fish went into which treatment, things started to make sense. When we go back here, that subadult was actually in this treatment tank. And what I think this is starting to tell me is that just being slightly larger and farther along in its ontogeny or its life history actually made it more willing to go farther and to eat more. It was less scared simply because it was larger, because it had more experience in its life, it was able to actually go out and eat more algae without having that extreme caution of the juveniles that we see in the control. So it's really cool to be able to identify something like this. It was maybe a happy mistake, an unintended piece of information we got out of this little trial, but nonetheless, it's going to help me in the long term because I now know that these small juveniles are inherently scared regardless of what we put into the tank. They're not going to travel all the way out to the farthest distance to eat simply because they're small and they have not had enough life experience to be able to make those decisions without having extreme caution. So super interesting finding, and I'm excited to be able to take that information to my next round. So I just want to briefly show you what this looks like graphically. Again, we have on the bottom here this treatment where I had the adult, and then you also have up here our control. So as you can see, once again, we had extreme feeding across the way with the individual that was kind of a subadult. We also had represented here, nothing happening in that control once again. But one more noteworthy thing about when we plot this graphically is that if you see here, even with that subadult, there was actually about a 2 to 3 day period where he wasn't very interested at all. So it almost shows that after about 2 days, 3 days, that subadult, unlike the juveniles in the control, was kind of able to flip a switch and say, you know what, these laminated groupers, I don't really think that they're actually a threat. I'm going to go start testing it out. And once he realized that nothing was going to happen, you started to see these really dramatic decreases in the algal height. So it's really exciting. It's not necessarily the question that I wanted to answer with this original trial, but it is incredibly important for me to tinker with this and to take away some really important information for when I set out to create my next step in this experiment. And on that note, our next step is just bigger and badder cage matches. Right now, I only have 3 of these set up. The long-term plan is for me to set up a total of 12. And once we get all of those going, we can alter all of these different densities of predators. We can alter densities of the prey. We could also look at what non-lethal predator interactions will look like. And hopefully we'll be able to uncover what is really going on with predator-prey interactions and how that influences herbivory. And ultimately, how does that play into what's happening with coral reef halos, this really mysterious and fascinating pattern that we see all over the world. So that's all I have. Obviously, thanks to everybody here in my lab, everybody at Moku'aloe who's been helping me, and obviously huge shout out and thanks to Sea Grant for their support. And hopefully you guys have some questions.
Thanks, Zach, for your talk. A round of applause for you. Let's take a look at the chat. I did see some questions coming in. Rachel, would you like to ask your question out loud?
Sure. I'm just sort of curious in the context of, even though it didn't happen, your other cage that you put 3 fish in. What were you expecting that was going to do? Would they be encouraging each other and be less fearful, or get in each other's way, or what was your guess that was going to happen?
Yeah, absolutely. So the original hypotheses were that I think in a situation where all of my fish were exactly the same size, whether we all had just all subadult or all juveniles, the concept is that no matter what situation, having more herbivores is going to lead to more herbivory, simply just out of numbers. But also, like you're saying, there's also power in numbers in terms of how they show anti-predator behavior. So the concept originally was, as I increase the number of herbivores and keep my predators constant, that halo or that distance that they're willing to feed should always be increasing. So basically, we want to understand and manipulate these different densities and see how we can expand the willingness they're willing to go, or in the opposite, increasing predator density, how can we actually shrink how far they're willing to go? So had the 3 actually stayed in there, ideally that would have showed that they ate a lot more than the group of 2.
Thanks, Rachel, for your question and Zach for your answer. What I'd like to do now is I'm going to bring back all the speakers to the Zoom stage. Let's see if I can figure that out. So let's get John and Matt back here and Becca as well. And if there are questions from the audience for any of the speakers, we can definitely take those right now. Okay, so I got our speakers back. Let's. Oh, go ahead.
I was curious about on your algae selection for your Manini. There's an awful lot of different types of algae out there. And I'm just curious how you chose the one you did.
Yeah, for sure. So it was a really long process. And for those that may not be as familiar with algae, this is by no means the Manini's preference. And the reason we went with this one in particular is basically it's the only one that was structurally sound enough and palatable enough to go inside of these clips and last for long enough periods of time for me to take these measurements over longer intervals. In a perfect situation, I'd love to design a setup where we have turf algae, what is their preference, and be able to measure that long term. But unfortunately, it's just too difficult to take accurate measurements over these time scales. But it's something I'd love to explore more. So it really just came down to, I know they'll eat it, and I know it will be structurally sound enough to last in these clothespins long enough for me to see something.
Okay, thanks for that question. I see a question in the chat for Matt and John. Let's see if I can find it again. It's from Beth Lenz. Beth, would you like to ask your question?
Yeah, it was just it's a silly random question for the issue that you're actually addressing, but for the electric resistivity metric where you're sending pulses through the sand, how does this differentiate from the calcifying macro invertebrates that are occupying that space?
Like how would it pick up the difference, or does it have an impact on it?
Does it have an impact? Does it create noise? Does it impact the animals? Do we care that it impacts the animals?
That is a great question, and I wish I knew an answer. I don't know. I tried to look up any literature about it quickly, and I couldn't find anything. Our guess is it doesn't, but there'd have to be more work specifically. It's not that high of voltage or like amps. I don't know anything about electricity, but I don't think we're sending that much electricity into the sand that it would have a large impact, but I'm sorry, I don't have a better answer.
No worries, just curious, thank you.
Okay, I see another really great question from Dr. Josh Maiden. Would you like to unmute yourself and ask your question? The question's for Zach.
Oh, hey, Zach. Yeah, I was just wondering, could the herbivores in your experiment have been using the roy as shelter after they realized that they were just kind of inert objects?
Yeah, it's a really interesting question. I think, had I let it run longer, I think we may be able to see that at a longer time period. This only ran for about 6 days. I don't think that was happening because when you actually look really closely at the data, in both scenarios, the control and the treatment, there was the least amount of herbivory was occurring at basically sites 3 and 4, which were actually the closest to the fake roy. So they were more willing to actually go to the farthest point than they were the closest points to the roy. So I think even if they were kind of, I don't think this thing's a threat, they still had a tendency to go to the points that were farthest away from it.
Oh, cool. That's really interesting. Thanks.
Okay, and I'd like to hear from Becca one last time before we close the seminar today. So I'll give Becca a question. In your talk, you had a call for action for all of us in the audience to go out and volunteer with these place-based groups. Can you tell us what that's been like and how that's informed your research, doing some of this stewardship and caretaking in the places where you conduct your research?
Yeah. So, I mean, obviously, pre-COVID, it was a lot easier to meet with community members. That was always very helpful in formulating research projects because they are really the community, particularly the stewards at HEIA that we work with, are sort of the first eyes on anything that we see, and they're very observant, and they will often identify things, see things that lead to further questions that we might be interested in investigating. It's also very important to have a give and take relationship. So just being able to sit down and talk story, have a conversation about the science that we're doing really goes to further trust for us in a lot of these areas, and allows us to have that kind of community access and access to their thoughts, their different beliefs as to what's going on in the system. And really, because they are the boots on the ground, they have a lot of experience that some of us, particularly graduate students, we don't have built up yet. So it's a great way to start thinking about your system from a whole new angle and perspective. Just by talking story. And then of course, it always goes to further a lot of ongoing further collaborations and research partnerships. It's sort of the gift that keeps on giving. So I would encourage everybody in their research, if you do research, to definitely go out and talk to the people that are most impacted by your work, and then also hear from them on their opinion on your science. It's always very good.
Thanks, Becca, for that answer. We're going to close the Science by the Sea seminar. We hope that you can join us next week. We have 3 new talks next week, so please come back Thursday at noon. Thank you to all of our graduate student speakers. Great job today on your presentations. And thank you to the audience for joining us over the lunch hour today. That's it, and we'll see you next time.